Myself, Dr. M. Sivaji, Teaching Assistant, Agriculture Biotechnology, Agriculture College and Research Institute. I'm working as a teaching assistant. Myself, Dr. M. Sivaji, I'm working as a teaching assistant in Agriculture Biotechnology, Agriculture College and Research Institute, ACNRI, Valachanur, Tiruvannamalai. I'm here to speak about regulation of gene expression and Operon concept. First, we will see what is gene. A particular segment of DNA or polynucleotides which contain genetic information to make final functional molecule, that is enzyme or protein, is called as gene. Gene is the basic physical and functional unit of heredity. The genetic information which is present in the gene is made up of DNA or polynucleotides. The particular segment of DNA or polynucleotide which contain the genetic information which is used for development of or producing of protein or enzymes, which is called gene. So genetic information flow is based upon the central dogma concept. Central dogma concept. The central dogma concept is put forward by Crick. It is explain the flow of information, how the genetic information is moved from DNA to protein. The DNA is having genetic information that is in bases, the amino nitrogenous bases, adenine, thymine, guanine, cytosine, like that. So the combination of these bases will produce some useful functional information that code for protein or amino acids. So in how the DNA is converted into protein, this explains the central dogma concept. First, DNA, the make copy is converted into mRNA, the mRNA through transcription, and the mRNA converted into protein through translation process. Normally, the prokaryotes, the DNA is transcribed into mRNA. The mRNA is translated into protein. That the transcription will happen in nucleus and the translation will happen in the cytoplasm. So here the gene expression is the genetic information is produced with the functional protein. The genetic information is from the DNA is converted into produce specific character that which encodes the particular gene which encodes the specific character is expressed or produced through transcription and translation process. This process called gene expression. Normally, any morphological character such as pod size, flower, stem length, number of panicles, this everything is determined by single gene or group of gene. So the expression of that particular gene will produce that particular character. So how the cell, the plant is having several parts. Every part is having same genetic material. The cell present in the every plant parts is having same genetic material, but the leaves are looking green. The stems are producing branches, and the branches produce flowers, and roots are in brown color. So the functions and the structure, color, everything is changed since the cell is having uniform genetic material. How the variation is happen. So this explained. This can be explained that the genetic regulation of gene expression. So normally gene expression is two types. One is constitutive expression. Another one is a regulated gene expression. The constitutive expression, the gene will be expressed always in all the plant parts, in all the cells, and from starting to ending of that particular plant life. That regulated gene expression will be expressed in particular plant parts based on the need. For example, the rubisco gene is expressed in leaf. The chlorophyll gene is expressed in leaf. That's why it produces, it looks green color and it synthesizes, it fixes the atmospheric carbon through photosynthesis. At the same time, the flower color is something different, not the green color, because of the anthocyanin and carotenoids, some other pigmentation due to that particular gene expression. So, based on the need, the expression of gene, the expression pattern will vary. Here, the prokaryotes and eukaryotes, two different organisms. The prokaryote is the basic model of the cell, and eukaryotes is advanced model. So the eukaryotes is having individuals organelles, and prokaryotes is not having well-developed organelles. So in this, the eukaryotic organism is having more amount of genetic materials that should be expressed. Minimum up to 30% of the genetic material will be only coding protein sequences. So the large genetic material is there. That's why it contains single gene will be regulated by single regulatory unit. So one promoter will control one gene. That also, the gene contains exon and introns. When the exons and introns here, that both are not transcribed into mRNA. In this, means the functional mRNA, the mature mRNA should have only exons. If introns and remaining things need to be removed, and it has to be modified by adding capping and poly-A tail for its expression. And in prokaryotes, it's very limited space, and the genetic materials also very less. So every genetic material, every base will act as a gene. That also, the more than one genes which is involved in a single pathway, single metabolic pathway will be grouped together in a cluster, and that complete cluster will be controlled by a single regulatory unit, because here the space and the genetic material is very less. So more genes can be controlled here, single regulatory gene. In case of eukaryotes, it's not like that, because the junk DNA and the introns are too much, since it has more genetic material. Now we will see control of gene expression in prokaryotes, because in prokaryotes, it's the gene regulation is first studied in advanced manner in prokaryotes, because the prokaryotes is having very less genetic material. There is no junk DNA like that. So detailed study has been done in prokaryotes. So approximately 4,000 genes in the typical bacterial genome. Only a fraction of gene expressed at a time. So there are two types, constitutive and regulated gene expression. Constitutive gene expression, as I told earlier, is expressed in all the cells in all the time, the lifetime of this organism, whether it is plant or bacteria. The regulated gene expression, it indicates only the gene will be expressed in particular the spatial temporal expression. The time will vary, and the place the expression will vary between the plants. So that is called regulated gene expression. The bacterial system is well studied about how the gene regulation is happening. The Jacob and Monod in 1961 proposed the model, that is Jacob-Monod model or Operon concept. In that, they explained how the metabolism of lactose and synthesis of tryptophan happen in the microbial bacterial system. Now we will see Operon. What is Operon? The operon is a set of genes or cluster of genes which are linked and transcribed together under the control of single regulatory unit or single regulatory region. The genes encode the particular function which are involved in a single metabolic pathway are clustered together. For example, the lactose metabolism, the lac operon which is involved in lactose metabolism is having 3 genes which 3 is important for lactose metabolism. And the trip operon having 5 genes, the 5 cluster, the gene cluster, it is having cluster of 5 genes. It all involved in tryptophan synthesis. So regulatory genes that also there in the operon that codes for regulatory, the regulatory gene codes for regulatory protein. It might be act as a activator or repressor that bind to the operator site to control gene expression of the operon. So different components of operons are regulatory gene, which codes for regulatory protein that control the regulatory protein that control the expression of that particular operon. So it may be activator or repressor. The operator site that is present in front of coding regions means structural genes and after promoter. So in the operator region, operator site, the repressor protein will bind, repressor or activator, the regulatory protein will bind and it will regulate the operon expression. The promoter site, the region where RNA polymerase need to be attached with the promoter, then only the transcription will start. So the promoter site is most important for transcription. Then structural gene, it's lays behind the promoter and operator site. It's having all the important protein coding or enzyme coding regions. So this needs to be transcribed if operator site is free. The structure of operon, the operon containing promoter, operator, lac Z gene, lac Y. It shows that image shows the lac operon structure. The PI is the regulatory promoted region. I is lac I gene. It's a regulatory gene which produce regulatory protein that act as a activator or repressor. So the operator is in between promoter and structural genes. How the prokaryotic gene expression is regulated? The RNA polymerase need to be attached with the promoter site. Then only initiate the transcription. The transcription initiation is regulated by the regulatory proteins, that is repressors or activators. The repressors, the name itself indicates it repress or suppress the operon expression. The repressors impair access of RNA polymerase to the promoter. It's in contrast, the activators, upon binding with the RNA polymerase, the transcription rate will be higher. So that's why it is called as activators. The regulation by means of activators, regulatory protein bind to the DNA and enhance the activity of RNA polymerase at the promoter site is called as positive regulation. Because the binding of activator into the operator site, it increase the RNA polymerase activity, but it enhances the transcription rate. That's why it is called positive regulation. So upon binding of activator, the transcription will be higher. In the repressor, in case of repressor, upon binding of repressor in the operator site, the transcription will be stopped. That's why it is called negative regulation. The negative regulation again classified into two types: inducible and repressible. So inducible gene expression indicates the gene product which is present will be more. The gene should be expressed when particular molecule is present in the system or circumstances. So that's why it is called inducible. For example, the lac operon, the inducer induces the expression of lac operon, and the lactose binds to the repressor protein and prevent the repressor from binding to the operator region of the lac operon. So the binding upon binding of inducer into the lac operon inducer to the repressor, the change in configuration, it can't be able to bind into the operator site. So the transcription start as usual. So lac operon functions only when lactose is present in the media and in surrounding environment for the bacteria. So this express the lactose metabolism of bacteria, because the bacteria is need carbon source. It mainly utilizes glucose as a sole carbon source. But when the glucose is not there, it needs some, or it it seeks some additional uh, carbon source. So here the lactose is the disaccharides. When break down the lactose, it produce two monosaccharides. This is galactose and glucose. So glucose is the first foremost option as a carbon source for bacteria. If glucose is not there, then only it will search some other carbon source present in the, whether it is there or not in the surrounding environment, like that. So thereafter it will start it metabolize and utilize that as a carbon source. So the transcription of genes generally blocked by a binding of specific repressor protein at a DNA site called operator. So dissociation of the repressor from the operator, it initiates the transcription. So that can be done by binding of inducer into the repressor protein or regulatory protein. The lag repressor dissociates from lag operator when repressors bind to its inducer. In case here inducer is allolactose, it's isomer of lactose. The second one is repressible gene expression. Here, the gene product that decreases when particular molecule concentration is high. It is that lactose, Operon is the catabolite pathway. It utilizes the lactose for carbon source. The bacteria will need to utilize lactose as a carbon source. So that is a need to be break down. So in presence of that lactose, that Operon will express. But in repressible gene expression, it's the biosynthetic pathway. When particular amount of product is more, then that production should be stopped. Then only the unwanted, the energy would be saved, because all the time the gene is expressed, it requires some energy for producing of some enzymes. Even though it is not needed in that particular time, if it is produced, the energy will be spent for the production of that particular enzymes, but there is no use of that enzymes, will be the waste of the metabolic. It will utilize the energy, so the bacteria will suffer for its further growth and development. Since the need when it required, that time the regulation of gene is mainly concerned the need of gene, that particular time only will be expressed. Remaining things it will be switched off. So in the repressible gene expression, the biosynthetic pathway of tryptophan, when the tryptophan amount is very high or the enough, no need of additional tryptophan. So that time that trip operon should shut off. So transcription should not happen. So the tryptophan act as a core repressor that stop the trip operon, so that the biosynthesis of tryptophan will be arrested. So this we will see later, the repressible gene operon, the trip operon and the lactose operon. Now we will see lac operon. There is a inducible gene expression type. So inducible type of gene expression. So E. coli expresses genes for glucose metabolism constitutively, but the genes for metaboling other sugars is regulated in sugar specific. So in the need, if media contain galactose as a sole carbon source, then the lac operon will be switched on. So lac operon consists of two regions. Normally operon consists of two regions. One is regulatory region and a structural region. The regulatory regions containing gene of regulatory protein, that lac I, promoter and operator site. This picture shows that these are the 35 region and 10 region is minus 35 and minus 10 is the promoter where the RNA polymerase bind. Then the next to this promoter, the operator site. The immediate next to the operator site, the transcription will start. So structural region consists recoding regions, beta galactosidase, lac Z gene, lactose permease, and the lac galactosidase permease, lac Y gene, and thiogalactoside transacetylase. Here, this lac Operon containing promoter that P lac I, then lac I gene. Though the promoter is for that lac I gene, the expression of this promoter will produce regulatory protein that used as a active here, the repressor. So normally this is the negative regulation and inducible. Actually, the lac of operator site is in between operator site and lac I regulatory region. The promoter is placed. So the RNA polymerase will bind in promoter and it starts. It crosses lac Operon and it starts synthesizing or transcribing these structural genes. So for that, the promoter of lac I gene should be active so that the lac I gene will be expressed and produce the regulatory protein, that is repressor protein always. The lac I gene produce lac repressor protein. The lac repressor has two functional domains. One domain is that binds to the DNA of the operator region. The another one that binds to the lactose, the inducer. So when in binding of the lactose, the configuration of the protein, the repressor protein will change. The lac Operon is regulated under negative regulation. The binding of regulatory protein stops the transcription. Repressor will stop the transcription. The repressor will be always active. The repressor protein of the lac Operon is always active. So when the repressor protein is produced in that particular system, it immediately will go and bind to the operator site. So once the operator site is bound with the repressor protein, then the transcription will not initiate. When the lactose is present in the medium or that particular environment, the lactose will act as a inducer and it will bind to the lactose binding domain of repressor protein and change its configuration. And because of this configuration change, that inducer repressor protein still can't not able to bind with the operator site. So in absence of repressor protein in the operator site, the transcription will start and produce enzymes, beta-galactosidase, permease, and acetylase, and that will involve to metabolize lactose into galactose and glucose. Then the glucose will be utilized. So this will be the whole function of lac Operon. So here the repressor and inducer combination decide whether repressor binds to the operator site or not. So in turn it decides whether that lac Operon is need to be expressed or not. So the functions, the three structural genes that is present in lac Operon, that's a beta galactosidase. It has two functions: breaking disaccharide lactose into glucose and galactose. The converting small fraction of lactose into allolactose, the isomerization, through isomerization. This allolactose acts as a inducer that will bind into the repressor protein or regulatory protein. The lactose permease or galactoside permease is mainly involved in transport of lactose. It increases the permeability of bacterial cell wall so that the external galactose will enter inside through lactose permease and galactosidase permease action. The thiogalactoside transacetylase is so far it's not well explained, but may be used to transfer COA group from acetyl COA, transferring of acetyl COA group. The function exactly not known. Then that image shows that outside the lactose present, that permeability is increased so that through galactoside permease, so that all the lactose will enter inside. Here, the beta galactosidase will break down into galactose and glucose and also isomerize through allolactose through isomerization. This whole image explains how the lac Operon is functioning. The top image shows the promoter and the operator site where the RNA polymerase, the RNA polymerase will bind into the minus 35 and 10, the operator region, sorry, the promoter region. The promoter, the next to the operator region, it will start. The RNA transcription will start from the operator region. So normally the lag operon is the inducible operon. It's coming under both lag operon and tripoperon is coming under negative regulation because the regulatory protein binds to the operator site. It arrests the operon expression. In case of positive regulation, the regulatory protein, upon binding of the regulatory protein, the transcription or the expression of that particular operon will be very high. So the binding of repressor protein arresting the transcription. So this is coming under negative regulation. In the negative regulation, the lag operon is coming under inducible type. So normally the regulatory gene will be always active. It produces through the regulatory protein that is called the repressor. The repressor is always active in lag operon. Once it produced, it will go and bind to the operator site, the next to the promoter where the transcription initiate. So upon binding of this repressor protein, the RNA polymerase will not move or will not cross the operator site. It will not transcribe that structural genes. So when it is the case when the lactose is not present in the media, if or surrounding environment of the bacterial culture. So if the lactose is there, then automatically the lactose will enter into the bacterial cell through galactose permease, and there some of the lactose fraction will be converted into allolactose. So the allolactose will act as a inducer when the allolactose is bind with the domain of repressor protein, the repressor protein configuration will be changed. So it's no more having ability to bind the operator site because of this structural configuration change. So after binding of inducer, the allolactose, the repressor protein is not active, that not able to bind into the operator site. The transcription is initiates. That's why it is called inducer. Normally in lag operon, the regulatory protein, that is repressor, is active. When it is produced directly, it will bind to the operator site and stop the lag operon transcription. In case of the presence of lactose, the small fraction, the allolactose will join or bind to the repressor protein and it change the configuration so that the repressor will not become inactive and it will not be able to bind into the operator site, and the transcription is initiates. So upon binding of inducer allolactose of repressor protein, that's why it initiates the transcription. Initiate the transcription. So this molecule allolactose is called as inducer. So the lag operon expression, how it is regulated. So both glucose and lactose can be used as a carbon source for E. coli, but the first preference is glucose. When the glucose is not there, then only the other sugars can be used as a carbon source. So there are three conditions. When glucose is present and lactose is absent, the E. coli does not produce beta galactosidase because the glucose is present. For that, there is no need of production of beta galactosidase. It will be waste. If the expression is there, that the enzyme and the energy it is used, everything will be waste. So the presence of glucose, the beta galactosidase, no need. The lactose metabolism is no need. So the lag operon will not be active. When glucose is present and lactose also present, the E. coli does not produce beta galactosidase because all the microbes, the bacteria, the first preference for its carbon source is glucose. So presence of glucose, till completion of glucose, this lactose metabolism is no need. So lag operon will be off. So when glucose is absent and only lactose is a carbon source, available carbon source of bacteria or E. coli, then now the beta galactosidase is most important. So like operon will be transcribed and it will be active. Here you see the DNA, the regulatory gene is produced, the regulatory gene is active and it produces repressor protein. The repressor protein is active when it is produced, so it binds to the operator site. Here the condition is lactose is absent. So in the absence of lactose, there is no need, there is no inducer. So its configuration will not, the change will not happen. So readily it will bind into the operator site. The operator site, upon binding of operator site, the transcription, the RNA polymerase will not move after the operator site. So the transcription of lac operon, the structural genes will be blocked. The lac I, the regulatory protein will produce the protein. The repressor, which the repressor is active and it will bind into the operator site. The RNA polymerase will bind into the promoter. So due to the binding of the repressor in operator site, the RNA will not move to the uh, structural gene. So the transcription will not happen. And the glucose and the lactose both are present. Still lactose is there. Some of the lactose will be converted into low lactose and will be act as a inducer and it will bind to the repressor protein. But it is not strong association since the glucose is there. So even though it is there and the transcription will be very minimum, but the binding will be not that much. The binding of the inducer and repressor protein is not that much strong. So again, it will bind. The reassociation will happen and it will bind again operator site. When glucose is absent and lactose is present, that time definitely the beta galactosidase is most important. So the lac operon should be transcribed. So the small amount of sugar allolactose is formed until it will bind the allosteric site. The inducer will bind, allolactose will bind the lactose binding domain of the repressor protein. So that the repressor protein configuration will change. It will not bind to the active repressor become converted into inactive repressor after binding of allolactose inducer. Then the promoter will start. The RNA polymerase present in the promoter will start move on structural genes and produce the polypeptides, which is necessary for metabolism, lactose metabolism. So it's a summary. Carbohydrate, that is glucose and lactose present, activator protein is not bound to the DNA. So lifted off the operator site, RNA keep falling off promoter site, so no transcription. Glucose is present and lactose is absent, not bound to DNA, the activator protein, and the repressor protein bound to the operator site, blocked by the repressor, there won't be any transcription. There is no glucose and lactose, the bound to activator protein is bound to DNA, the bound to operator site, blocked by the repressor. The glucose is not there, only the condition lactose is present and bound to DNA, lifted off the operator site. The repressor protein will move, it will not bound to the operator site. So in the RNA polymerase will sit in the promoter site. So transcription occurs. Now come to the tripoperon, that is a repressible gene expression type. Here, this also explained in the E. coli. The tripoperon contains aromatic amino acids, which is important for protein synthesis. So the tripoperon contains 5 structural genes. In case of lacoperon, 3 structural genes, and tripoperon containing 5 structural genes, trip E, trip D, C, B, and A, which encodes the enzymes needed for biosynthesis of tryptophan. The 5 structural genes are next to each other, in which altogether called as tripoperon. So it also contains a repressive regulator gene called TRPR. The TRPR, the expression of TRPR will produce the repressor. That repressor actually, so this image shows the structure of and trip Operon. The regulatory gene is there, promoter is there, operator is there, in between the attenuator. So it's the control of expression, transcription. This region is utilized for control of expression, the leader sequence. And this image shows the gene expression and the function of the 5 structural genes. Here the TRPR, that is a regulatory gene producing a repressor protein, actually it is inactive. In case of lac Operon, the repressor which is produced through LAC-I regulatory gene is always active. So immediately after production, it will bind to the operator site. But in case of tryptophan, that is biosynthetic pathway, the repressor protein which is produced from regulatory gene is inactive. So it is inactive, it will not bind into the operator site. So the RNA polymerase which bound into the promoter, it will start the transcription since the operator site is not having the repressor protein. It will start synthesizing, producing all the polypeptides or the amino acids, that is TRPE. This gene code for anthranilate synthase. TRPD codes for anthranilate phosphoribosyl transferase. TRPC codes for phosphoribosyl anthranilate isomerase and indole 3 glycerol phosphate synthase. TRPB tryptophan synthase B protein and TRPA codes for tryptophan synthase A protein. So this all the polypeptides, the enzymes is involved for biosynthesis of tryptophan, aromatic amino acids. So expression, the transcription of this particular Operon will lead that the peptide formation through translation. Then it is involved to synthesis of tryptophan. When tryptophan is enough in the system, the tryptophan production is enough, then no need of tryptophan further. Then that Operon should be shut off. So in this case, the excess tryptophan, when it is more than normal amount, then that will act, that will bind to the repressor protein, inactive repressor protein or regulatory protein. Then upon binding of this inactive tryptophan to the repressor, the inactive repressor become converted into active form. So once it is active, then it will readily bind to the operator site. So the repressor, active form of repressor, once bound to the operator site, the RNA polymerase will not move from the promoter to the structural gene. So the trip Operon expression is getting arrested. Here see the configuration. Normally mRNA from transcription of LAC, that TRPR genes, the regulatory genes, that mRNA is translated into protein, that is repressor. It's inactive. It's not able to bind to the operator protein of operator site of the trip Operon. When the tryptophan is binding with the repressor protein, then it starts the configuration rearrangement. So it's readily bind to the operator site. So the repressor is inactive. Upon binding of tryptophan, it is become active. That's why it is called as co-repressor. In case of lac Operon, the operator site is bound with the normal active repressor. When the active repressor is bound with the inducer, that is the allolactose, it starts the transcription. That's why the allolactose is called as the inducer. Here, the binding of tryptophan will arrest the transcription. That's why it is called a co-repressor. Thank you.